Welcome students. Now we are going to start with a new chapter. The name is Useful and Harmful Microbes. As once again we will begin with the name and its meaning, and then we will go into the details. So this unit has a very important word. This chapter has a very important word called as microbe. Now let us go into the details of this word microbes first. Microbes word has originated, or you can say is a version of a word which is very well known to you. Microorganism. Now microbe is extremely tiny or extremely small microorganism. That means a body or a living body, you can say, which is so small that cannot be seen with naked eyes. So if it is cannot be seen by naked eyes, how do you think scientists must have studied it? Yes, you are right. Scientists have used instrument called as microscope, which you all must have seen in your school, or if not, you will see that in your later years. So microscope is an instrument which is used to see those tiny living organisms which you cannot see with your naked eyes. Now I will talk about microscope and how do we use it a bit when we go into the details of this microbes and its observation, its structure and so on. So when scientists started observing microbes, they realized that they are very much like any other living things with few exceptional features. Most of the times microbes are unicellular. That means one-celled. Sometimes they are multicellular. That means more than one cell. So they have cells which are connected to each other and they form different shapes. However they are, whether they are unicellular or whether they are multicellular, ultimately their size does not grow so much that we can see them. They are always in a size in microns. In microns, a micron is the unit that is used to count the smaller measurements. So or smaller lengths, you can say. So they are in micrometers or microns. So we cannot see them with our eyes. If we talk about more or less how do they look or what are their features, is I have already told you the first feature, that they are unicellular or multicellular. Now consider a microbe which is unicellular, then definitely it is not going to have organelles or parts, organs like us, but it will have some special organelles within itself which helps that one-celled microorganism to survive. For example, some microorganisms which are unicellular, they develop different shapes so that they can move. So they vary in shape so that they can move. They have different types of cell organelles which make them complete. They do not need multiple cells. So the cell shape will be such that they can move. Then the cell membrane, or you can say the cell wall, whatever it has, will manage it in such a way that it will change its shape to take the food in. Then the food will be digested, whatever fragment of that fraction of that food it will take will be digested within cytoplasm or another cell organelle. So everything is managed within cell. Similarly, even if the microorganism is multicellular, these multicells, the number will be obviously very less. They manage all the life processes that are required for the microorganism to survive. Now this is overall about microorganisms. In this unit particularly, we will be talking about few useful microorganisms, that is microbes, and few harmful microbes. And we will be covering their structural details, that means how do they look, what are their body parts, how do they support, how do they live. Then we will talk about its relation with human. How are they helpful to human? Now when we say useful, we are very much limited how that microbe is useful to us. And when we say harmful, we will be limited to how that particular microbe is harmful to us. Right? So let us now begin with the first useful microbe, which is called as lactobacilli or lactobacillus. bacillus is plural, is singular, and if you say CILLI, then bacilli, that is plural. So lactobacillus or lactobacilli, you can say, is the first microbe that we are going to study, which is useful to us. So I have a picture. You can now focus on the slide, and you can see there is something greenish, and then there are these magenta, or you can say reddish pinkish colored bodies. They are nothing but lactobacilli. Now this picture is taken the view under microscope. So sometimes when they take a photograph, the colors vary. Okay, so when I will explain how are these lactobacilli cultured, or the slide is prepared, the color that I am going to say is bluish red. This is not exact that color, but as I told you, the color may vary because of the shot taken, or sometimes the flashlight that falls on it, but more or less it looks very similar. So if you want to really take or make the slide of this lactobacilli and observe it under microscope, here is what you have to do. You have to take a little amount of either buttermilk, or you can also take yogurt or curd. You have to take few drops on the cover slip on the slide, and then spread it well, and then add a cover slip. So the slide is somewhat like this, which is a glass slide, and cover slip is another glass, very thin glass sheet, which you have to cover, you have to keep it on the slide. So here you will take the drop of buttermilk or yogurt. Why buttermilk is preferred? Because it is liquid, so it spreads very easily, and then you will cover it with the cover slip. But before covering it with the cover slip, you have to add one chemical, and that chemical is methylene blue, which makes it reddish blue. Okay, now this chemical is called as stain. Now what is stain? Stain is the chemical that is used on different, there are different types of stains. This is one of them, methylene blue. So when they are added to the organism that we want to observe, or whichever part of cell that we want to observe, that part gets colored, and then it becomes very easy to see. Because usually what happens is, when we take a drop of something or a very thin section of something under microscope, usually it is colorless, or it is maybe you can say pale yellow, pale green, so everything appears very same. But when you add these stains, or when you add these chemicals, what happens is, certain part which we want to observe takes that color and becomes colorful. So it becomes very, very easy to recognize that part under microscope. So to observe lactobacillus, what stain are we going to use? Methylene blue. And because of this methylene blue, this color is chosen, is absorbed or taken by the bacillus bacteria, and then you can see them very carefully. Now whenever you use microscope, you know that microscope has lens, right? It has two lenses, and a combination of these two lenses will give you proper magnification. It is eyepiece lens and objective lens. One is on top and one is at bottom. Now you have to take one lens as 10x. You can take any, you can take eyepiece lens which is next to your eyes as 10x, and the objective lens which is little down. If you want me to show you how it is, suppose this is the microscope, this is one lens and this is second lens. So this is called as eyepiece lens, which is next to your eye, and this is called as objective lens, which is near slide, and your slide is over here. Okay, so whenever this lens, both the lenses can be replaced. Okay, you can put whichever power lens you want. So one is 10x and one is 60x. So total, if you want to see how much magnification is that combination going to give you, you simply have to multiply, and the answer is 600. So in a way, if you want to understand, if you see an object of 1 millimeter, this magnification will make it 600 millimeters for you. So that 1 millimeter is that tiny, and 600 millimeter is almost this much. So that is how it magnifies the image for you. It's the image, it's virtual. actually the organ is not going to get bigger, or actually that slide is not going to get bigger. It's the image that gets resolved, or you can say there is a magnification. So at 600 magnification you can actually see this lactobacilli. Now if you look at the structure, they are more or less rod shaped. So they are rectangular and rod like. So if you take a rod and if you cut it into smaller and smaller pieces, how will that look? You see the structure carefully here. You can see this rectangular part and it's like a rod, rounded, right? So that is how lactobacillus look. So there are many angles these lactobacilli you can see in this picture, and where they are is called as substratum, means the material in which they are inoculated or they are present. There is one more important property of lactobacillus is the way it respires, or the respiration process, which is very crucial again for this organism, because breathing or respiration is essential for all the living organisms, because using oxygen or whichever way producing energy is extremely important. So you know that respiration, when you use oxygen and do it, you call it aerobic respiration. But when you don't use oxygen and you do it, then it is called as anaerobic respiration. Now anaerobic, N. Now this N makes it opposite. That means without oxygen. So whenever this lactobacilli are there in any of the material, they will not need oxygen to grow. They can grow without oxygen and they can produce energy without oxygen. But then what do they need? Even if they do not need oxygen, what do they need to produce energy? This, for anaerobic respiration, they need sugar. You also know that sugar is the input required for energy. Similarly, this lactobacilli will also need sugar. That is a mandatory thing. That is something a lactobacilli will always need to grow. So if you see this name of lactobacilli, so lacto, that is one type of chemical which is present in milk. So these bacteria or this lactobacillus is highly active in milk. Whenever you add this bacteria in milk, they start their process, that is anaerobic respiration, and do something. And as a product, what gets formed is curd. Now when lactobacillus form curd, what happens exactly is what we are going to study next. So until now what we have studied is what are lactobacillus, what are the main features of lactobacilli, and now we are going to study its action, what it does exactly when you add them in milk. Now when you say milk, milk has two main things in it. Milk has proteins and milk has fat, and of course milk has water. There are few more things that are there in milk, calcium and all, but we will not mention that right now, because our focus is what happens to proteins when you add lactobacilli, because lactobacilli affects, or you can say work on proteins. What happens when you add lactobacilli in milk is these proteins starts getting clustered, they start getting together. So suppose you have milk. In milk you have big fat globules. I am just showing it a pictorial representation. So the big ones that I am drawing and which are darker are fat molecules or fat globules, and the small dots I am making are proteins. So this is how they are randomly mixed in milk. So they are together in milk. The moment you add, I am just making it thread like just for you to understand. When you add these lactobacilli, this lactobacilli get mixed in here and they start separating these dots that are proteins from the other part of the milk. So what happens is the other part of the milk, the fat, water content and everything starts getting separated from this protein, and these protein molecules will start coagulating, means they will throw the things a little away and protein molecules will come closer. So after a while, after some time, it's not a very fast action. After some time, what you see is these proteins are actually forming like this. They are coming together and forming lumps. So you can see all these proteins are getting closer, and fats are there of course on their own. They have nothing to do with this process majorly. So they are there, but proteins will coagulate. They will come together. The word I am using is coagulation. Means they will form clusters, throw water away, throw fats away, and they will come together. So this is how they will coagulate, and once they coagulate, it will form a fragile solid. Fragile in the sense, you must have seen curd or yogurt. It looks solid, but if you put spoon in it or your finger in it, it is soft, right? It's not very hard solid. That's why it is called as fragile solid. So it makes a fragile solid because of coagulation of protein molecules, and that's how curd is formed. I am sure you must have got clear about the curd formation, because this is very important to understand the further uses of lactobacillus. Now, why this coagulation? I have explained that the coagulation happens and the protein molecules come together and all other things go away. Why this happens? Because lactobacilli will use milk sugar, will undergo anaerobic respiration, and as a product they produce lactic acid. Now once I say word acid, it has to do with a chemical term pH, that is percentage of hydrogen, and you all know that the percentage of hydrogen, that is pH of acid, is less than 7, right? So milk usually has pH that is more than 7, but because lactic acid is formed, the pH drops down to the lower pH, and because of which the coagulation starts and the protein gets condensed, or you can say coagulated, and the curd is formed. But this process is extremely important for you to know what exactly happened at that as a chemical reaction, and because of this lower pH and lactic acid formation, the taste of milk also changes. What it becomes? Sour. Now just to explain this to you, I will give you one example. You must have seen your mom making curd at home, because not always we buy curd from outside, right? But to make curd, you need curd. You cannot make curd without having some amount of curd with you, right? Why does that? Because that curd that you are going to add in milk has lactobacillus in it, and from somewhere you need that lactobacillus culture. So when you take suppose a bowl full of milk and you add say half spoon of curd, little curd, and you mix it, and what do? You keep it away. You keep it away for several hours, not only 1 or 2 hours, but you keep it for one night or sometimes at least 8 hours. If the climate is warm, then it is for 5 to 6 hours, but if the climate is cold enough, then it is overnight. You have to keep it for 12 hours. Why so? Now this is little outside your textbook, but try to think, why does this happen? Because warmer temperatures support faster coagulation, faster lactic acid formation, and faster curd formation. But if the climate is cold, like the temperature is below 25 degrees or so, this whole process slows down. So the curd formation also slows down, right? This whole process of curd formation is also called as fermentation. So lactic acid formation, because of which the curd is formed, is called as fermentation. I think you all must be clear with why sour taste, why slow or fast process. and what is fermentation. Now, sometimes what happens when you make a curd and suppose you have kept it in one corner and suppose you forget for 2 days that it is lying it in that corner, and then if you take it and if you try to taste, what happens is the curd has become extremely sour. And now by understanding the process, will you be able to answer this question? Please give it a moment and think about it. Yes, you are right that sour taste goes on increasing because of excess lactic acid formation. Excess lactic acid formation will make or will produce excess sour taste, and that's why the curd becomes so sour that you cannot eat it, right? But that curd is the best medium to make new curd because it has lot of lactobacillus. So one drop of that curd will be enough for you to make the next curd, right? So this is overall about the curd formation. Now I would like to talk about the medical part of it. Why is curd of medicinal use? You must have heard at your home, like when you have stomach upset or you have stomach ache or pain, ache, your mom tells you कि बेटा थोड़ा कर्ड खाओ या कर्ड के साथ राइस खाओ या कर्ड के साथ या सीधा कर्ड खाओ, थोड़ा शुगर मिलाओ और कर्ड खा लो. क्यों ऐसा बोलते होंगे? Why do you think your mom says that? Now, if you want to really think for the reason, please think about this. When you eat curd, it lowers down the pH, and very low pH is not suitable for harmful bacteria. Okay? So when you have stomach problem, of course you have stomach problem because you have some infection in your stomach, right? You have some microbes in your body that have gone in. That is why you are getting that pain. So if you eat curd, what curd will do? Curd will reduce the pH, because of which, like there are many bacteria who are causing this pain will be killed because of lower pH, and then your stomach issues will go down slowly and slowly. Now, one more thing of medicinal use I would like to tell you, that in market nowadays you must have seen this, and nowadays you get flavored yogurts also. Yogurt is nothing but curd. So you get yogurt which is plain like curd, or you get mango flavored, strawberry flavored, and you get it in nice colored small portions you can buy, right? And sometimes it is written probiotic on some of the food products, or sometimes on this also. What do you mean by probiotic? Probiotic is few microorganisms that are added to the food product so that the food product becomes more healthy, more helpful. Now as you know, this lactobacilli are perfectly helping us in better digestion or to kill the microorganisms. If these are added to some other food items, of course they will also become more useful, you can say, or more healthy. They will make the food item, milk food item, more healthier. That is what is probiotic. So these are added to other milk products also so that they become more helpful. Let us talk about the uses now. So this whatever I have talked about is also the use of lactobacillus, but there are some more uses of lactobacillus. The first one is of course in the milk product formation or production, sorry. Now milk product, when I say, it's not only curd. By making curd which has cream in it, you also make ghee, you also make different other items that are like ghee, butter and all. So white butter or the butter that you get ready made in market, the original product is curd. So that is how milk products is the main use of lactobacillus. Secondly, cocoa, pickle, usually pickles that are made up of vegetables. Because if you want something to sustain for a longer time and get that particular sour taste, you know that pickle has that sour taste. So that sour taste is due to lactobacillus. So pickle formation, production, or cocoa for production, you use lactobacillus. Then one more use which I have already explained you is medical. Different types of medicines which are nowadays called as probiotics are with the help of lactobacillus, because they kill the microorganisms that are harmful and make it more useful for the person, and the pain goes down very fast. One more important and little different use of lactobacillus is fodder for cattle. Now you know that cattle usually eat hay or dry grass, and that's how they fill themselves. But if you want to make this product more or their food more healthier, you can add lactobacillus. And what do? Usually they take this hay or dry grass and they add a little bit of water. They inoculate means add and keep lactobacillus overnight or for few hours. They process it, and then that cake is given to the animals to eat, so that it has lot of microorganisms that if they go in their body, they also make their digestive system more healthier. So fodder for cattle, and sometimes it is used in wine and bread making. Bread making, lactobacillus is not the major microorganism, but there is one more that we are going to study afterwards. But it is some types of breads that are used with lactobacillus, and wine, because you know that the basic process for wine is fermentation. So any fruit wine when you make, you take the fruit, you inoculate it with the microorganism and you ferment it. Okay, so that is how what we have studied until now is features of lactobacillus, uses of lactobacillus, and action, how it actually works.